Good morning for everyone. And the title of my talk is T-cell responses to cancer. But as Sophie highlighted, I'm going to be talking also about chronic viral infections because what my lab is really interested in is in T-cell differentiation, and this dysfunctional state that we see in cancer can also be observed in other situations. What I want to talk today is about T-cell activation. And I think the first talk today was very important to help me introduce this topic, because when we think about T-cell activation in cancer, we need to think about the steps that we usually think about what a cell needs to be primed. And the first thing that a T-cell needs is really to recognize the antigen. So in cancer, T-cells can either recognize tumor-associated antigens or tumor-specific antigens. And there are some little differences in terms of abundance and affinity. And I think we are just now beginning to understand, and there are groups at Sinai that are really contributing a lot to this field. And what are the differences when a T cell recognizes a tumor-associated antigen, maybe has lower affinity, maybe has seen it before in a more tolerogenic form, but when they're really tumor-specific, and they're usually much higher affinity, but then there is a problem of abundance sometimes. So I think those are really interesting topics, and this is very important for T-cell response to cancers. And of course, the only way we can have a T-cell response to cancer is if the T-cells can see something in those cells that are different than they see in healthy cells. So this is really a key part. But we cannot forget that as important as it is to have TCR and peptide, T-cells cannot get activated if they don't receive co-stimulation, cytokines and chemokines. And I think there is a fourth component now that we are aware, and especially inside the tumors, there is the nutrients in the microenvironment. And for example, there can be a lack of glucose, there can be for example, excessive lipids or a lack of lipids, lack of oxygen, and all of this can influence T-cell differentiation. And this is as important as initiating a response as it is later to reinvigorate responses. So I hope by the end of the talk, I can convince you that a lot of the steps that we usually think about how to initiate a response are also very important when we want to reinvigorate anti-tumor responses. And sometimes in cancer treatment, what we are going to do is try to reinvigorate tumor-specific cells that are already present. And in some patients, unfortunately, those cells are not there. Then they need to be either generated, for example, with vaccinations, or other strategies that have been used is actually adoptive transfer or making cells that are not tumor-specific being able to recognize cancer cells. But I'm not going to be addressing this extensively today. So my lab, what we are really interested in is in T-cell differentiation. And I just mentioned that for a T cell to be properly activated, it needs to receive not only TCR signaling, but it also needs to receive other kind of cues to be able to really expand and become an effector cell that's functional, that can kill. So the problem with cancer is that a lot of times we think about the tumor microenvironment and unable to provide those other cues. So the effector stage might not be as pronounced as what we are used to, for example, in an infection. But still, a lot of the steps that lead to T-cell dysfunction in cancer are very similar between cancer and other situations, such as chronic viral infections. But what happens in cancer, and in chronic viral infections, which is really key to the state of T cells that we usually encounter infiltrating tumors, is that we find exhausted cells, and it's the persistence of antigen. Okay, so this is the clear difference that this, for me, and what people have shown, the features that we find when we find T cells that are infiltrating cancers, the one that are tumor specific, what they have is signs of antigen persistence, of chronic antigen stimulation. And I want to highlight also there has been some newer studies that have been emphasizing that right from the beginning, when you generate this initial effector response, there are already some precursors of cells, and those precursors can either give rise to those memory cells if the antigens are clear, or they will be able to give rise to those cells, the exhausted cells that are able to persistently survive in this environment of antigen persistence. So there are very interesting steps in the beginning in this priming event that I'm not going to be focusing today, because usually when we find a patient with cancer, those T cells have already been primed. So this is the state that I'm going to be dealing during this talk. So T cell exhaustion. So T cell exhaustion happens through persistent antigen stimulation, and it's a progressive dysfunction. So the longer you wait, the worse those cells become. So even though the dysfunction can happen very, very early, epigenetically, this becomes progressively worse. So if you take a cell that has been chronically stimulated for a couple of days, for example, and you take it out from antigen stimulation, it will recover and it can become a memory cell. And if you re-stimulate, it can become a functional cell. However, if this cell is stimulated for a long time, then it's really hard to recover. And also, it becomes worse and worse, for example, in terms of cytokines. So some cytokines are lost very early on in this process, for example, IO2, whereas interferon gamma can last quite a bit. So this differential program that we call exhaustion involves epigenetic, transcription, and metabolic change. And it leads to T cells that have reduced ability to proliferate and produce cytokines. However, it's important to realize that those cells still have some functionality. So they are dysfunctional, they are hypofunctional, but they're not inert, okay? And the other thing that's very important is that they become dependent on antigen to survive. So unlike memory cells that survive, they self-renew due to cytokines, those cells become addicted to antigen, and they need antigen to survive. And those exhausted cells have this very unique transcriptional landscape that's induced by the transcription factor tox, and they have very high levels of several inhibitory receptors, including PD-1. Okay. And so I'm going to spend just one slide talking a little bit about PD-1. And PD-1 is this inhibitory receptor that's expressed due to TCR signaling. And that's very important because that means that every cell, when it gets activated, will upregulate PD-1. But usually when an effector cells upregulate PD-1, the antigen is gone, then PD-1 that gets downregulated. But when cells are chronically stimulated, PD-1 becomes higher and higher, and then other inhibitory receptors are also expressed and contribute to this hypofunctional state. So PD-1 binds those ligands, PD-L1 and PD-L2, and when those ligands are bound, it gets phosphorylated and recruits SHP2, which can then dysphosphorylate downstream signaling of CD28 and the TCR. Okay. So this is how PD-1 is an inhibitory receptors, and it's, even though not all the mechanisms of PD-1 inhibition are elucidated, not even on T cells, and also PD-1 can be expressed in other cells, and that gets a lot more complicated because we don't really understand what it could be doing in other cell types. But other inhibitory receptors expressed by T cells are not as well known, the downstream signaling as PD-1. So this has been very successful, but we all know that even though it has effects in many different cancer cell types, because it's such it's a shared mechanism of immune regulation that the T cells are trying to fight those tumors that are expressing new antigens. But we know that patients that receive those therapies, only a small proportion can respond. So this is the problem of the field. Everyone is again trying to understand why are only some patients responding. What can we do for the other patients that are not responding? And that's a little bit what I'm going to be talking right now. But the way we address this is really trying to understand in the patients that do respond, what are the key steps that are happening that allow those responses to happen? Okay. So as I talked about it, so if we have this exhausted cell and it's receiving negative signals because it expressed PD-1, and tumor cells and antigen-presenting cells can express PD-L1s. Well, so can we take away those negative signals and reinvigorate T cell responses? However, we knew that when you're first mounting an immune response, naive T cells, for them to expand, in addition to receiving TCR signal, they also need to receive positive costimulatory molecules, and for example, through CD28 expression. So the question that I started addressing back in my postdoc, which I'm going to revisit quickly just to introduce the topic of my lab interest, is really, is co-stimulation also important beyond priming when you're trying to reinvigorate those PD-1 positive CD8 T cells? Okay, so I focus first on the CD28 molecule, which is expressed on naive cells, and it's also still expressed in some subsets of exhausted cells. And CD28 binds to B7 and B7 ligands, which are usually expressed in professional antigen-presenting cells, which is a very interesting concept, because in the few years ago, and there are still some people nowadays that when they talked about PD-1, they think about PD-1 and PD-L1 acting only at the tumor side. However, and I hope I will convince you by the end of this talk, because even PD-1 blockade need to have positive co-stimulation. Well, and because PD-L1 is expressed also by those antigen-presenting cells, and the CD28 signaling is key, I think what the concept that we have right now is that PD-1 is also very important at this APC T-cell interaction, and not only at the interaction between tumor cells, okay? And that's a major regulation of the immune response, is at this stage of T-cell interaction. Okay, so the basic question, are positive signals required for T-cell rescue? So a few years ago, we were able to address this question and show that for PD-1 target therapy to be successful, we need to receive CD28, the T cells need to receive CD28 signaling. And we showed this using this chronic viral infection model of lymphocytic meningitis virus. We use tumor model and supporting data in lung cancer patients. So quickly, we had cells that recognize the virus, this LCMV virus that are called P14. They were either wild type here or CD28 knockout. And what you can see is when the chronic infection is established, you can detect those cells in the blood. However, if you give anti-PD-L1 blockade, those virus-specific cells which express PD-1 and are in this dysfunctional state, they can expand. And you can see this T-cell expansion here in the blood, actually. However, if those cells lack CD28, they were unable to expand when you gave anti-PD-L1, okay? But CD28 is very important for the T-cell priming. So we also wanted it to be able to take away CD28 only after T cells had undergone priming. So for this, we also used those transgenic cells that were specific for the LCMV virus, but the CD28 was floxed and it was going to be deleted only when CRE is expressed, and we use CRE ERT2 mice so that when the CRE is expressed only when we give tamoxifen. So those virus-specific cells were transferred into mice. Mice were chronic infected with LCMV, so that you have those cells in this exhausted state, which is very similar in chronic viral infections and in cancer. When chronic infection is established, we can give tamoxifen to delete CD28 on those virus-specific cells, and then we give anti-PD-L1. And what we could see here is that proliferation measure here by Ki67 expression was really restricted by the cells that express CD28. And the cells where CD28 gene was deleted, we could stain here by flow cytometry here, and those cells could not proliferate. So you can see here that all the cells that are proliferating are the cells that express CD28, and the cells that lost CD28 were not proliferating. And I think this is also a very important concept, that this CRE-RT2 is not super efficient, but we are able to stain for CD28 and really look at the cells that have deleted and really analyze what's the difference between the cells that have deleted and the cells that haven't. Okay, so this was an eosinophilic chronic fire infection, but we also wanted to know if the CD28B7 pathway was important for reinvigoration of tumor-specific cells. So we used the CT26 colon carcinoma tumor model. And what you can see here is that in untreated mice, tumors can grow. If we start treating mice that have tumors around day 12 with anti-PD-L1, most of the mice, the tumors regresses very nicely. You can see here this little curve. However, if together with this PD-L1 blockade, we block CD28 interactions with B7 by giving anti-B7 antibodies, the tumors cannot be controlled by anti-PD-L1 treatment, showing that also tumor-specific responses need re-engagement of the CD28 for this anti-PD-1 therapy to work. And we also had the opportunity to look in cancer patients. And at that time, it was probably when I started this experiment, maybe even 10 years ago, looking at TILs from patients. We were still trying to understand this diversity of T cells, and we were surprised to see that even though most of the CD8 T cells in non-small cell lung cancer express PD-1, when we looked at the CD28 expression, there was a lot of heterogeneity between the patients. So in some patients, 80% of the PD-1 positive cells were CD28 positive, but in some patients, only 20% of those cells were CD28 positive. But then patients that were receiving PD-1 blockade, we can measure the cells that respond in the blood. So we could see that in about 60 to 70% of the patients, when they receive PD-1 blockade, if you look 2 to 3 weeks later, you can see that PD-1 positive CD8 T cells start to proliferate. And here again, looking by Ki67, the cells that are proliferating. And when we look at those cells that are proliferating in the blood after PD-1 blockade, we could see that most of those cells were expressing CD28. So there was this disconnect between the phenotype of the cells that were in the tumor microenvironment and the cells that were proliferating in the blood after therapy. And at the study, that's just the observation that we could have. And so we proposed that this CD28 interactions would also be important in patients, but that's the extent that we could go at the time in the study. But we could conclude that in animal models that it was very important for CD28 signaling for those CD8 T cells to respond to PD-1 blockade, implying that this pathway, the CD28B7 interactions, would be very important for effective PD-1 directed immunotherapy. So in the last few years, this T cell exhaustion and heterogeneity of T cells has become a lot more clear and also maybe more complicated. But what we now know is that what we were calling like those PD-1 positive CD8 T cells are actually a mixture of cells. And there are many ways that we can subset cells even further and further, especially with single cell RNA technologies. But for the purpose of the talk and the framework that I would like to understand, I want to talk about 3 different kind of PD-1 positive exhausted cell. And one of them is this progenitor, healthier TCF1 expressing PD-1 positive CD8 T cells that I'm going to be talking about TPEX, and some people call them memory-like or stem-like. And those they express less inhibitory receptors, even though they express PD-1 and they have tox and they have a lot of the chronic antigen stimulation signature. They express overall less inhibitory receptor and more positive costimulatory receptors. So this is a very important subset that I think really changed the way how we think about T cell exhaustion. So, and this is a small subset among those PD-1 positive CD8 T cells. And then there are two kinds of cells that I would like to frame them like that. There are cells that are more effector-like, and this is the one that will have more functionality. This is the state that we want to expand when we do therapy, when we want to reinvigorate those cells. That's the state that we want to expand. And then there are the really terminally differentiated cells, which are really the more dysfunctional state. Okay. And what we learn about the dynamics of those cells is that those progenitor exhausted cells, they self-renew and they differentiate to this effector stage and also this more terminally differentiated stage. And whether it always goes like this or there is a bifurcation is still a matter of debate. But the important thing is that those progenitor exhausted cells are the cells that are giving rise to the other states. Okay. And here, again, the transcription factor TCF1, which in RNA or gene is TCF7, and those other more differentiated states express CD39 and TIM3, for example. So what I want to go back a little bit is to our findings about CD28. And when we looked more carefully at those different kinds of PD-1 positive cells, what we saw is that the progenitor exhausted cells were the one that expressed really high level of CD28, whereas the more differentiated cells had less expression of CD28. And this was also true when we looked in patients. And if we look at cells that do not express TIM3, but that express PD-1 in non-small cell lung cancer tumor-infiltrating CD8 T cells, the cells that do not express TIM3 have higher level of TCF1, and they also have higher level of CD28. So it seems that, and now we have studies showing, including a study that I'm going to show a little later today, that by single cell technologies, we can identify those subsets that we've seen in animal models. We can also identify them in patients. So our working model for how this is working right now is that those progenitor exhausted cells are interacting with professional antigen-presenting cells, and our collaborative studies and other group studies have shown that those progenitor exhausted cells can be found in draining lymph node, but also in the TME in close proximity to those professional antigen-presenting cells, and we think they're receiving CD28 signaling. And this is important because when you do PD-1 blockade, they need the CD28 signaling to differentiate, okay? So what we didn't know is how is CD28 signaling regulating this differentiation of those exhausted cells? And what we saw is that when we have mice that are chronically infected with LCMV, if we treat those mice with antibodies that block CD28 and B7 interaction, there is this drastic reduction on virus-specific cells. You get that can be identified by tetramers that identify virus-specific cells, and there is a reduction in lymphoid organ and non-lymphoid organs. And there is, because I told you that those dysfunctional cells have still some functionality, if you actually reduce the number of those cells during chronic viral infection, you have an increase in virus, okay? But what was very surprising to us is that when we looked at those subsets of progenitor exhausted and the more differentiated subset, what we saw is that those two subsets were equally affected by this B7 blockade treatment. So when you take away CD28 interaction, we saw a reduction in the progenitor exhausted cell and in the more differentiated cell. And when we did that, and we then are able to sort those progenitor exhausted cells that either express high level of CD28, intermediate levels of CD28, or no CD28 expression, and we label them and transfer them to infection-matched mice, and we looked at their differentiation potential. What we see is that only when the cells express high level of CD28, they are able to differentiate. And you see here, they were all TCF1 positive when they are transferred, and they start expressing CD39. And what we can see here is that when we quantify the potential of differentiation of those cells according to CD28 level, you can see that they need CD28 signaling to be able to differentiate. Okay, so what we concluded is that differentiation of those progenitor exhausted cells is regulated by the level of CD28 signaling, with differentiation requiring more CD28 signaling and self-renewal a little less. So when we try to understand what is CD28 signaling doing in those cells, we saw that the oxidative phosphorylation was affected when cells lost CD28 expression. And what we saw, we did some seahorse experiments to measure the metabolism of those cells. You can see that oxygen consumption rate of those progenitor exhausted cells that have lost CD28 is completely it really ablates their ability to do oxidative phosphorylation when they lose CD28. We also measure mitochondrial with mitochondrial ROS, and what you can see is when they lose CD28, we really lose this mitochondrial ROS, and we also did EM and saw that when cells lose CD28 expression, their mitochondria become very disorganized and unhealthy. So we really think that CD28 signaling is regulating the mitochondria also beyond priming. So those cells need to continuously be receiving CD28 signaling at least a little bit to keep a healthy mitochondria. However, to differentiate, we know that effector cells need glycolysis. So we measure how those different levels of CD28 signaling would promote glycolytic activity on those cells by measuring acidification rate in the media. And what you can see here is that the more CD28 signaling, the more glycolysis those cells can do. And we confirmed that CD28 was modulating the level of this transcription factor IR4, which has been implicated in effector differentiation. So what we did also, we inquire whether if we can increase CD28 signaling by giving a CD28 agonist, if that will make the cells, those progenitor exhausted cells, more functional. And we measured this by transferring those progenitor cells either coated with a CD28 agonist or untouched into mice that were LCMV chronic infected. And what we can see is that the CD28 agonist improved the differentiation of those cells. And what was very striking is that they express this molecule CX3CR1, which is usually associated with those more functional effector-like cells. So I want to go a little bit quickly here, but we also showed that when those cells received the CD20 agonist, they were better able to control tumor growth. Okay, so this was published last year in Science Immunology, and what I hope I can convey here is that CD28 signaling is still controlling those cells beyond priming, and this is through metabolism. And it's not just all of nothing, but the level of CD28 signaling is very important, and you need a higher level of CD28 signaling to induce more glycolysis, and that's what's going to give you a better effector-like differentiation. Okay, so I want to take the last few minutes of my time here to shift gears and show our data in patients, a collaboration with Miriam. And I think it's very important that we try to link the mechanistic studies that we do in mice with what we observe also in patients. And in this particular trial, we had 25% of response rate that was measured as tumor necrosis. So when we look at the patients that received PD-1 therapy, we could see patients that had a lot of T cells, patients where the T cells were excluded from the tumors, and also patients where the tumors were quite low for T cells. And not surprisingly, all the patients that responded to PD-1 therapy, they were classified as T cell rich. They had a lot of T cells in their tumors. But what was very surprised that an equal number of non-responder patients also actually had a very heavily T-cell infiltrated tumors. So we want to understand what are the difference between those T-cell infiltrated tumors that respond and those that do not respond. I'm going to just go a little quick. So we did single cell RNA sequencing of those CD8 T cells, and we were able to identify the same subsets that I've described earlier in mice. So we could identify different states of PD-1 high CD8 T cells, and those PD-1 high were all enriched in the tumor compared to adjacent. It's very important to have an adjacent to compare. And they all had this signature of chronic activation, but we could identify cells that were had this more progenitor phenotype, including TCF1 and XCL1 and other molecules, and as well as this more effector-like subset. And when we quantified the frequency of those PD-1 high CD8 T cell states, what we could see is that the cells that had this more effector-like were enriched in responder patients. Okay. And what was very important for us is that we could also use the TCR sequence as a molecular barcode to understand those different states of those cells that were expanded in tumors. So we looked at the phenotype of the top 10 tumor-enriched clones for responders and non-responders. And what you can appreciate is there is a lot more orange in the responders. So those cells are in this more effector state. Whereas in non-responder, you have more green, they're in this more terminal state. But what was very important for us is that those progenitor cells here, they were present in non-responder as well, okay? And they were also in responder, but they were also present in responder. So that for us, when we saw that data a few years ago, was quite surprising because in animal models, it's been shown that those progenitor exhausted cells are the cells that respond to PD-1 therapy. And if they are in non-responder patients and we gave PD-1 blockade, why didn't those cells expand? Why did they look more terminal rather than effector? So we tried to look at other cells and other that could influence T cell differentiation in the tumor microenvironment. And what we observed is that there was a correlation between having those effector CD8 T cells that correlated with tumor necrosis and CD4 cells that express CXL13 in the tumor microenvironment. So those CXL13 CD4 cells, they also have signs of chronic activation, which have some feature of follicular helper cells. They also have some Th1 phenotypes. And these subsets were also highly enriched in tumor. So, and other studies have shown that the CXL13 is a really good marker for tumor-specific cells. And the CD8 T cells that were PD-1 high were also CXL13 positive, which is quite interesting. So, so it seems that patients that respond to treatment, right, had also those CD4 cells in their tumors, whereas the patients that were T cell enriched that did not respond to anti-PD-1 therapy, that had a much lower frequency of those CXL13 positive cells, and actually they have if you measure their makeup of CD4 cells, they would have more Tregs, which is also very interesting. So another important question to understand how those interactions, we design probes that would bind to the TCR of those really expanded clones so that we could visualize those cells, not only post-therapy, which is what I have shown you so far, but we also had pre-tumor biopsy. And I'm just going to blow up those dots here so that everyone can see them. What we saw is that those clones that are highly expanded after therapy, they were already present in the tumor microenvironment before therapy, okay? And when we look at the clonal expansion of those tumor expanded clones, we saw a difference between responder and non-responder in the tumor, but we did not see a difference in the draining lymph node in those HCC patients, okay? So this data support that even though there are progenitors in the lymph node, that this expansion and differentiation of those CD8 T cells is likely occurring in the tumor microenvironment. So we wanted to understand what else could be in this tumor microenvironment that could be leading to this expansion. And I realize I only have a couple of minutes to finish here, but I want to link with the two previous talks that we had today. And this is Miriam's beautiful work on dendritic cells in cancer patients. And she recently identified this MREG-DC, this state of mature DCs that are enriched not only in maturation molecules, but also some of the immunoregulatory molecules, such as they're pretty high for PD-L1, for example. And those cells can be identified by expression of LAMP3 or DC-LAM. So when we looked at those cells in tissues, what we could see is that around those cells, we could find those progenitor exhausted CD8 T cells, as well as those CXL13 CD4 cells, and they were forming these triads. And when we quantified those triads, the proximity of those cells in responder and non-responders, those triads were more enriched in patients that respond to therapy than in patients that did not respond. Okay. So what this study that was published in Nature Medicine last year is proposing is that in patients that respond to PD-1 therapy, those progenitor exhausted cells need to interact with this MREG DCs and also need to be in close proximity to those CD4 cells expressing CXL13. And those signals then are able to trigger, we think, this effector-like differentiation. Whereas in non-responders, maybe those cells are not interacting with those dendritic cells, maybe because they don't have those CD4 cells to guide them. And then those cells, when you give, they give rise to this more terminally differentiated subset that is not as functional. Okay. So I just want to, this is my last slide. Just want to go back to the first slide, because I think a lot of the things that we study and we think about when we activate naive cells are also very important when we want to reinvigorate. But there is a difference, especially if we want to reinvigorate. We are dealing with, we're not dealing with naive cells anymore. We're dealing with those progenitor exhausted cells, which have a different set of costimulatory molecules, chemokine receptors, cytokine receptors, and different needs in terms of nutrients. So I think it's very important to understand what do those cells need to be able to be reprimed and differentiate to this effector stage, which I think that's what we need to be able to better control tumors. And of course, if patients don't have T cells that recognize tumor antigens, then other strategies will need to be employed, such as tumor vaccinations or giving T cells or really changing the whole myeloid compartment to really start to have and maybe kill some tumor cells to liberate more antigens to generate those T cells, okay? So I just want to end thanking my group, especially Etienne Humblin, which led all those studies in LCMV. And this, I showed a lot of my collaboration with the Mera Lab, Asaf, Pauline, and Regeneron. And I'm happy to take questions.